And Adarsh, the
Yeah,
floor
okay.
is yours. Thank you.
Yeah. Good morning, everyone. We have Dr. Dilantha Fernando with us today. He's a plant pathologist, a professor, and dean at the Department of Plant Sciences, University of Manitoba. He's an internationally renowned host pathogen interaction researcher, specializing in epidemiology and the spread of pathogens in canola and cereals. His core research area is sustainable agricultural systems and climate resilience through environmentally friendly disease management practices such as resistance breeding with genetic traits, biological control, microbiomes, AI, and research on plant growth promoting rhizobacteria or PGPR. He has been associated with naming resistance genes in canola and introducing new disease management strategies in Canada. He's a frequent keynote speaker at conferences and sits on several boards nationally and internationally. He's a fellow of the Asian PGPR Society, the Canadian Phytopathological Society, and the American Phytopathological Society. He has won numerous international, national, and local prestigious awards for research, teaching, and services to the community. In 2023, he received the Lifetime Achievement Award for Excellence in Agriculture and Environmental Sustainability. In 2020, he received the award for CPS Outstanding Research, and in 2019, he received the highest award for excellence in research on PGPR. Professor Fernando is the founding editor-in-chief of Plants MDPI and sits on the editorial boards, various other editorial boards worldwide. In January 2023, he was appointed as the editor-in-chief of Plant Pathology, which is the flagship journal of the British Society of Plant Pathology. We are delighted to have him here with us today to talk about the genetics of fungi and host plants. So over to you, Dr. Fernando.
Thank you very much, Adash, for that introduction. You can see, if you can see the slide, the first slide, can you just tell me that you can see that so that I am not worried
Yeah,
about?
Yeah, it's
Okay.
good.
it's
Yeah.
good.
So I have changed this title. I thought this might be a better title for this presentation, particularly because it's really a success story, and this particular success story has now been accepted as a research article in plant pathology, and it will be coming out very soon. It's in the proof stage right now. So after you listen to my presentation, and if you want more information as to how we got about doing the research on understanding evolution of fungal pathogens to come up with the canola-blackleg interaction and reduction of this disease, you are very welcome to do that. So that's part of the reason that I changed the title so that it matches for the audience that I'm speaking to. So thanks again to the Evolution of Fungal Pathogens group for inviting me to give this talk today. And one of the things that I thought I should quote here uh, is that the world needs massive numbers of people to cooperate with one another to make progress. And it needs dreamers to weave well intention stories to spark our collective imagination. Stories are the greatest human invention. People need stories to cooperate. Now, this is very appropriate because we have a very young audience of graduate students and postdocs here, and they are the real dreamers, and they are the people who carry out the research. We also have uh, professors like Dr. Richard Hamelin, who has already dreamt of all this and made that imagination come true. So, and my intention here is with the presentation today, if there's an opportunity for some of you to see the value of collaboration with my lab on some of this research that we are doing, I will be very happy to talk to you about it in the future. I was visiting China for work last about in July and August, and one of the plugs caught my eye, what it said, one seed can change the world. One technology can create a miracle. So if there are any Chinese, you can see whether I'm telling the truth here. This is a quote from President Xi Jinping. And I think today's talk is absolutely in that very direction of one seed changing the world, and how technologies that we have used have created disease mitigation. So just to give you a little bit of background where I am at the University of Manitoba, the oldest university in Western Canada, which started in 1877. We have a student population of about 35 to 37,000 students in a given year, with undergraduate and graduate students, almost in all the disciplines that can be at a university. Now, University of Manitoba is also quite famous for the crop that I am going to be talking about today, canola, which was developed in the Department of Plant Science where I work. And the reason that it is called canola, for those who may not be aware of this word, is it comes from Canada plus oil. And it is a crop that is now the #1 cash crop in Canada. Now, the question that I can ask the audience is, are we on the right track? We have come a long way in our evolution. And again, we are talking about evolution of fungal pathogens. We are doing extremely well in some of the work that we do, but sometimes on our own selves, we haven't particularly paid much attention. So there is a real need for us to get back on track to make sure that we use zero trans fats and to be healthy, so that we have a healthy population that is not going to be an issue for the health industry and the governments. Now, on that note, canola sits at the very top, with the saturated fats being the lowest in canola oil, and some of the other polyunsaturated and monounsaturated oils that are so important for health are in high numbers in the canola oil profile. Now, as I mentioned, canola was first developed at the University of Manitoba, Department of Plant Science, by Professor Baldur Stephenson, who is in this picture. And today it is almost a $30 billion crop grown in Canada each year. Now, the University of Manitoba and the canola program was recognized with a Canadian stamp. And I'm very happy to put this up, even though it's now about 15 years old, because this was, I'm not sure how many research programs have been recognized by the Canadian government with a stamp. So my research is more on the pre-breeding, finding the right genes, and also working on 3 major diseases: blackleg, verticillium stripe, and Sclerotinia stem rot. So some of the work that has been done by my lab has received several awards, and these are 2 awards that I have just mentioned here. Now, canola, Canada is one of the best known countries to grow canola, but at the same time, you have to recognize that there are many other countries that grow canola or rapeseed, and those are the countries that are shown in this slide. Now, one of the key things to remember is that canola is grown in over 22 million acres for about $29.6 billion crop each year, which tells us that there will be issues coming up when you have a crop as good as that. And we will talk about the issues that come along with it. But one of the key things that we need to remember, Canada has a population of about 40 million, and we grow much more canola than what we can consume. So over 90% of the canola grown in Canada is exported as oil, seed, and meal to markets around the world. So there is a real need for us to make sure that what we do in canola in research or mitigation of diseases are all done not only to provide a high-yielding crop, but also to meet the world standards some of the countries that might ask for. And as I said, a world leader in canola, where we have 31% of the global production, 69% of global seed exports, 65% of global oil exports, and 69% of global meal exports. So when it comes to such a large and such a important crop, unfortunately, there are issues that come along. And as I said, one of them is Leptosphaeria maculans, the fungus that causes blackleg in canola. Now, blackleg is a very interesting pathogen. As you will see in the presentation, we have learned a lot from this pathogen and a lot that may not fall into the textbook style of genetics. Now, Leptosphaeria maculans can cause disease on the seedlings. The lesions can come up in the leaves, stems, and also girdle the stem base. You can see these pycnidia production on the leaves or the stem, and then they mature and produce more spores for the growing season. Now, as I said, it is a pathogen that can infect almost all parts of the plant. And here on the very right-hand side, you see the pseudothecial production, the pathogen's pseudothecia production shown on the right-hand side. That is on overwintering stubble that they will be producing that. So if you look at the global distribution of the pathogen Leptosphaeria maculans and a lesser aggressive Biglobosa, you see that the pathogen can be, both of those species can be present in several places around the world, and some places will have one or the other. And in China, you have only L. Biglobosa. So there is and China is a huge importer of our canola, and we need to meet some of these standards that they might need or impose on us. We will get to that in a short while. Now, getting to the classical gene for gene theory, Dr. Harold Henry Flo introduced this through his work on the flax rust system. And I always sometimes think that some of these excellent researchers who have done so much for our understanding of any particular system were very lucky, because when he worked, it was a fairly clear system on the flax rust system where a gene for gene theory could be developed. He used the flax rust. I won't get into this. You are familiar with this if you are a fungal biologist. I will just go into what the genes would have been looked at from his angle of the flax rust. So if you have a R gene and an AVR gene, R gene in the host and the AVR gene in the pathogen, there has to be a match. It's like a lock and key. You cannot take somebody else's key and open your lock into your office. So the same thing is true when it comes to pathogens. There has to be a lock and key, or in scientific terms, we call it effector-receptor interaction. So this particular thing that was introduced by Dr. Flo is not particularly something that is being followed in the host pathogen interaction of canola and blackleg. We will come to see that very soon. Now, in the classical gene for gene interaction, this is in canola that I have shown, you will see that single R gene will interact with its corresponding AVR gene. That's the lock and key mechanism that I was talking about. And you will get the interaction to give a hypersensitive reaction. In all other cases, you will end up with susceptibility or disease. So there will be pycnidia production and much inoculum will be produced because the pathogen is able to cause disease. So for example, a capital R interacting with a virulent allele will give disease, and so is when it is a recessive allele of the host interacting even with the AVR gene will give you the disease. So what we learn from this is that not all genes, R genes and AVR genes, interact the way that we learned in the textbook. There are genes that will we call play hide and seek or hijack genes that will have an impact on the phenotype. Phenotype is what the farmers really see. It's not the genotype that they see, it's in their field, what they are really encountering is the phenotype. So there is a greater need to study these economically impacting interactions and evolution of fungi to make agriculture profitable. Now, here's a picture that was taken way back in 2011 when we started to see that R genes, varieties that were labeled as R genes, were not really working as R genes. So in the field, what you see when you see the phenotype, you see a lot of disease on this R-rated canola cultivar. This means that there is something happening in the field that makes that resistant variety not act as a resistant variety. So when we bring that into the lab and do cross sections or look at the stubble, you will see that it is almost like a susceptible plant. It is not showing any resistance. The pathogen has caused disease enough to show this blackening effect in the stem base. So getting back to the types of resistance we have, even in canola against blackleg, we have two types. That's the major gene or qualitative or vertical resistance that is carried out by single genes, and the other by minor genes or quantitative resistance that we encounter, which are having small impacts but collectively can have a very good impact on reducing disease. So if I had to put some pictures to what I just showed, you see that when there's no resistance, you will see disease on the seedlings and also on the stem base. When you take horizontal sections, if there is major gene resistance, you will see that there is resistance in the plant with hypersensitive reaction, and also there will be a very clean base when the genes are working. And when the minor genes are there, even though you might see some seedling susceptibility, you will see a much cleaner stem base at the end of the season. So that is quantitative resistance. So both these resistances are important, but when it comes to breeding, it is much easier to identify and breed for qualitative resistance rather than quantitative resistance. So again, reminding you with this slide that I had put, it is a gene for gene interaction that we hope for, and this is what the pathogen will initially show if there is a effector-receptor interaction. Now, so in 2011, when we started to see that resistance was breaking down, there was an opportunity for us to look into this with collaborative research with several different scientists across Canada, and also with scientists from France and Australia who have had more experience with the breakdown of resistance in the past. And so we collectively came together to develop research projects that would now be carried out in Canada to look into this particular breakdown of resistance. So to do that, we had to use to characterize the R genes, we had to use the different pathogen isolates, and for characterizing isolates, we had to use differentials of the host canola plant that we knew of the R genes. So here's a snapshot of the differential Leptosphaeria maculans isolates that we used for R gene characterization. And sorry, and you can see that different isolates carry different amounts of AVR genes. For example, D3 will carry AVRLM5, and also, just go ahead. Okay, AVR-LEPR1 and AVR-LEPR1. D4 will carry AVRLM4, AVRLM5, AVRLM6, AVRLM7, and so on. So with this kind of isolates where there are more avirulent genes than a single avirulent gene, you have to do a deduction method when it comes to identifying the correct R gene. Okay, so this particular slide shows what we were able to find after about 2 years of work, where we were able to find, we were able to see that R genes in the Canadian canola germplasm was not something that we expected. Most, whether it came from Monsanto, Bayer, BASF, Syngenta, you name it, Cargill, most of them carried RLM3. So this immediately told us why we were seeing a breakdown of resistance in the Canadian canola cultivars. The work was done by one of my PhD students at the time, Jiangshu Hua. And what we noted was that there was a lack of good diversity of our genes in our genotypes that were grown in Canada, or the elite germplasm that was developed. At the same time, we were interested in profiling the AVR pathogen population in Canadian farmers' fields. And to do that, this is how we collected the pathogen, and we carried out the work with a master student that I had at that time, Sakraya Liban, who looked at using differential cultivars that carry different R genes so that we can characterize the AVR pathogen population. He was able to show that the most predominant AVRLM3 was the virulent alleles. The orange that you see here on the third column is or third bar is the virulent allele. So which means when virulent alleles are at about 97%, that is one of the reasons that you see this very that strong resistance of RLM3 being broken down, because there is no gene for gene match. There is 97% of the alleles or the pathogen population carries the allele for the virulent allele. So there will be lots of disease in the field. So the phenotype that the farmers see is not exactly what we would expect from the classical gene for gene interaction. So this is where we were able to match the two sides of the work, and all these are published, so you are very welcome to take a look. But as I mentioned, the article that will be coming out in Plant Pathology in about a month's time will give you a very good abstract version of all what I am presenting today. So so this is exactly what then the farmers will see, even though they bought the R-rated variety from the retailer from to make sure that their variety will be safe under disease pressure. Unfortunately, that is not what is happening in the field. Now, here's the bad news in a cartoon style so that you understand. So when the AVRLM3 interacts with RLM3, what you get is the classical hypersensitive reaction, that is resistance, and that is good news. But unfortunately, when there's a virulent allele, what you get is not exactly a hypersensitive reaction, but a susceptibility where there's lesions coming up in the variety. So this is exactly what is happening in the farmer's field. Now, there's even more bad news when it comes to some of these genes having other genes in the pathogen isolate. So for example, if the AVRLM3 has also the AVRLM47 as a virulent allele, the resistance is shown and a hypersensitive reaction is shown. Now, as we saw in the previous slide, when the pathogen has the AVRLM3 as a virulent allele, even what the AVRLM47 showcases doesn't really matter. There will be susceptibility. However, the next one is the important one, where when AVRLM3 is as an avirulent allele, when there is in that same pathogen isolate AVRLM47 as a avirulent AVRLM47, there is susceptibility shown as the phenotype. So unfortunately, so this is what we can call aggressive mimicry. The presence of a functional allele of AVRLM47 hides the RLM3's mediated recognition of the AVRLM3. So even though at the genetic level, this is showing to be like a gene for gene interaction, a hypersensitive reaction, it is hidden because of the presence of the AVRLM47. So it's a evolutionary new trick of the pathogen to overcome the host defense responses, and this is a epistatic suppression of the AVR recognition in this particular gene RLM3. Now not being able to be shown as a RLM3, but more like a recessive gene because of the presence of the AVRLM47. So one of my postdocs looked at this from both angles, did greenhouse research, bringing all these isolates and trying to look at the phenotype, as well as did genotyping to look at whether AVRLM3 was present in most of these isolates. And what we found was that over 80% of the isolates from the field carried the AVRLM3, the avirulent AVRLM3. However, in the greenhouse, what we see as a phenotype was very low of the presence of AVRLM3. So which means that there is a mismatch of the PCR, that's the genotyping, and the phenotyping because of the masking effect by AVRLM47. Now, this game of hijacking and hide and seek perceived credentials was further characterized with another AVR gene by one of my postdocs, Kaveh Ganbania, and working in Dr. Hossein Bohan's lab. We were able to show that RLM9 was also the AVRLM9 presence to give a receptor effector interaction was masked by the presence of the AVRLM47, the same gene that we were talking about with the AVRLM3. And again, the same situation happened. The opportunity for the presence of AVRLM9 was masked by the presence of the AVRLM47. So again, there was epistasis and the suppression of AVR recognition. This was also looked at more carefully, looking at all the locations that we could look into where the SNPs were and all the genes were by failure. And again, it was very similar to the masking of the AVRLM3 by AVRLM47. This time it was on AVRLM9 by AVRLM47. So I'm not going to go into the discussion about epistasis. This is a slide just for you to understand that epistasis can happen not only in pathogens and plants, but also in humans. Now, in theory, I put the word theoretical. What you will expect is this kind of interaction where AVRLM1 will interact with RLM1 and so on. But unfortunately, that's not the case. And there are many situations of the combinations of AVRLM3 and AVRLM47 that can mimic or mask the genes and create an opportunity for the pathogen to overcome that resistance in the field. So this is showing how PCR would show and how the greenhouse test. I will just concentrate on just a couple here. So here you see the AVRLM3 presence with PCR. You see the genotype, but unfortunately, it's not present when you look at the pathogen on the plant, because the plant is particularly showing the phenotype, and that is because it is being masked by AVRLM47. So how does this affect the canola cultivation? As you can easily see, it is going to be hugely impacting the canola cultivation because the gene for gene interaction is not particularly happening. There's hijacking and hide and seek happening in most of these cases, especially because RLM3 was the gene that was most predominantly available in all the canola varieties that are grown, in most of the canola varieties that are grown in Canada. So this was the case until about 2017, 2018. There's much good news after that, but let's get into that slowly. So what can we do about this? So there's lots of things that we can do. The first thing that will come to your mind if you are a plant pathologist is to recommend rotation with non-host. Now, will that work? Of course, it'll work, because there's lots of crops that can be grown that are not crops that can be infected by blackleg pathogen, Leptospira maculans, and it will be very easy to do that. However, it's easier said than done because farmers are looking at the high cash value crop of canola to be what they would want to grow. And that's one of the reasons that I am a firm believer that we need to do certain things to meet the requirements of the farmer rather than dictate to the farmer by saying something like what I have right here. This is true, this works, but unfortunately, this may not be what the farmer requires because, for example, barley might bring a very low amount of money as returns to the farmer, whereas canola would bring much more. So they would rely on growing and taking a risk with canola because of that reason. So if you are not a plant pathologist, you will appreciate this slide, even though this is a little bit of an exaggeration that I am making. So if the canola was grown in the first year and you go into a tight rotation, if the pathogen was just present in just 2 locations in the field, in your field, now it can manifest into much more inoculum because you have given the same kind of variety that to be grown in the field. Now, if you give a third year, that's really tight rotation, the pathogen might be uh, in your whole field where the pathogen might be able to infect. So this is how the pathogens virulent races develop, and a whole area can be devastated because of the virulence of the pathogen. Now, as I mentioned, rotations do work even when it is a susceptible variety. You can see the reduction of the pathogen as you go with a 4-year rotation away from canola. And this is work done by Dr. Randy Kuchar in Melfort when he was in Melfort, Saskatchewan. And resistant varieties would be really good in a rotational system. However, as I said, that is not the case when it comes to the economics of a crop, and most of the farmers might take a risk. Now, this risk was very well evident in Australia in 2002 when a company developed a variety and called it Silvestris resistance, or even called it complete resistance to blackleg. And when the crop was grown in 2002, almost all the farmers in Australia who grew canola grew that same variety. And because that was a very good variety with high resistance, they tried to grow it the next year and see what happened in 2003. So this is what we call the boom and bust cycle of any particular crop when a host and a pathogen interacts. And this is what the pathogen was able to do, overcome that resistance that was there, the Silvestris resistance, and cause major disease. So we are trying our best to avoid this now that we understand that there is a breakdown of resistance happening in Canada. And some of the methods that we have used, or the labeling systems that we have used in Canada, will be shown in the next slide. And the following slide, I will show you what, as a group of scientists, we have come together and ask for new labeling system so that we can avoid what is happening. So in the good old system, what we rated, what we called in the rating was either a resistance, moderately resistant, moderately susceptible, or susceptible. This is the way that the Western Canadian Canola Rapeseed Recommending Committee agreed upon, and for years this was followed. This was rated with the susceptible variety Vesta as a check, and the percent was given as such. But we knew that this was not a good one, because if the farmer bought a R-rated variety and if it didn't work as R-rated variety, what's the use? So we came up with this labeling system where we, to make it easy for the farmer to identify what kind of resistance they might rely on, we came with resistance A, B, C, D, E, and each of these labels of alphabet, a letter in the alphabet, meant that it carried a major resistance gene. And each of these then would make sense because now, as I said, when we did the initial research in 2011, 12, 13, we found that RLM3 was the only one that was present in predominantly. But today we have the luxury of having more resistance genes because of the work that was done in those 3 years to showcase to the industry that we were lacking that diversity. Now they have gone through and bred for resistance with other new genes. So we asked the companies to come up with the labeling, not only with the R label, but also tell what R gene they carried. And most of the companies went ahead with that. And they, when you buy a variety now, you can look at whether it is carrying RLM3, RLM3 and RLM4, or RLM3 and RLM2 and RLM4, and so on. So now we have an opportunity, RLM3, LEPAR3, and RLM5. So we now have an opportunity as a farmer to make that decision well based on pathogen population that you might have in your field. So to do that, for example, if you had high incidence of the pathogen presence in your field in, let's say, 2017, in 2018, I have put this particularly 2017 and 2018 because that's the year that we came up and this strategy of new R gene rotation was introduced. And R gene rotation has now been successful for several years now in reducing the disease. So for example, if there was disease with the gene labeled A, that is, let's say, RLM1, you will not grow RLM1 resistance in the next year. You have other options. So you can try other options. And sometimes where there are 3 genes might work, but see that there is also RLM1 in that particular mix. So, the farmer can now make an educated decision. Now, this was shown very well in work done in Australia as well, where when there was the disease in the previous year in group D, and the next year they grew group D cultivar. This is what happened. The pathogen was able to overcome that resistance. Whereas in that same, with the same stubble, group D stubble, if you grew group E, you will get a very healthy crop. So this was something that we were proposing that we should do in Canada. Now we went further with the work Kaveh did, my postdoc did in Dr. Hossein Bohan's lab. He developed cast markers to clearly separate out the pathogen profile in a farmer's field. So now these cast markers are available with the seed testing laboratories, so that these seed testing laboratories can do for a nominal fee for the farmer to identify the particular type of AVR profile that they have, and decide which RLM gene or RLM genes will work in their favor for the next coming seasons. So this is what is followed by different crops, seed companies. Here's one example of Bayer Crop Science calling that traditional R, but also putting ACG, so that the farmer now knows that if he buys RLM1, RLM3, and RLMS variety, whether it will work or not based on the AVR profile that we had identified for them with the cast marker development. So from the understanding we had of R genes and the pathogen population in Canada, it was proposed that R gene labeling should be adapted in Canada. And now I'm very happy to state that it is a practice and it is being carried out in almost, except for one company, almost all the other companies are carrying out this practice. So I'm also a firm believer that data by itself is useless unless you can turn it into recommendations to use on the farm. This was said by Dr. Rob Fraley. I think he was the CEO of Monsanto at one time at a Grow Canada conference. And then to see them in use is really gratifying for a researcher. And particularly, I mentioned this to the young researchers, it is important that you publish well, publish in high impact journals, but at the end of the day, your work that you do in the lab, greenhouse, and in the field has to end up with recommendations to the farmers. So this is another clear indication of what I've been just mentioning. RLM3 on RLM3, this is Canadian research. You'll see that the pathogen can easily overcome the resistance in the field and cause havoc. Almost all the plants are diseased. And RLM4 grown on RLM3, that same stubble here, is an excellent crop. So knowing that, we came up with validating of our genes with research undertaken by my master's student, Justine Cornelson at the time. And she worked very closely with farmers. And to make a long story short, what she identified was that there were many genes that would work very well. However, I'm presenting here that something that you need to remember, that is, even with two genes, sometimes the genes that are in combination really matters. So here you have RLM3. If RLM3 is alone, there's lots of disease. You can see that. But when RLM3 is with RLM4, the disease is reduced heavily. So you can see this again. I'm putting that same slide again for you to understand that RLM3, the RLM4 protects the RLM3 from being causing disease as much as RLM3 alone. And what we did then was to bring all these varieties to the same field and do the work in one field. And again, we saw the same result, which was very interesting. This is also published material, that the RLM3, RLM4 combination, whichever cultivar, so these are two different cultivars, but they had very significantly low disease compared to the RLM3 alone with different types of cultivars. So as you can see, with the evolution of the pathogen, what we learn from each of these stages is very, very interesting. So the good news is that with the work that we have done with collaborators, we have been able to clone the AVR genes. We have been able to clone the R genes. That was done in Dr. Hossein Bohan's lab. And also come to understand how actual Leptospira maculans interactions do occur. So not every AVR gene is interacting with one R gene. Sometimes it so like AVR-LM1 interacts with RLM1 and LEPR3 and so on. So these have been confirmed with transgenics, and there's some genes that are allelic. You can see, again, these have been all published, so I'm not going to spend a lot of time because I have a few more slides to finish. There is sometimes the epistasis that I was talking to you and gave you examples of, the AVR-LM3 and AVR-LM9 being masked by AVR-LM47. And then there are digenetic genes where pathogenic variants in two different genes are required for a particular condition or trait to be present. So all these can be happening at the same time. Some of these genes are at the same locus. So what we have learned from the actual Leptospira maculans interaction is absolutely interesting. It is not a simple gene for gene interaction. There's much more to it that keeps for us to keep finding. So there's much more work that we are doing with collaborators, that we are finding each day something new. So I'm going to skip that and just show the pathogen races that might be available in a particular region. So this is characterizing all the pathogen isolates across the prairies, Manitoba, Saskatchewan, and Alberta, and you can see how the pathogen is well spread. And just in Manitoba, you can see the pathogen profile of the AVR alleles. Now, what are the key findings? What we have been able to let the farmers and the scientific community know is that our gene labeling works, our gene rotation works, and how to overcome some of the key predominant avirulence alleles or the virulent alleles that are present in a diverse pathogen population that is uneven. So you can ask the question, if the our genes are not labeled, what will happen? So that for that too, we have an answer. Because you remember we had developed these CAS markers. So all that you have to do is do, so far, here's a good example. This was work done during COVID. We went and collected samples in Alberta, and so from a field that had not had a variety that was labeled. I told you a particular company was not labeling the varieties. And what we get from the report that is developed after the genotyping is done by the seed testing laboratory, we can predict what RLM gene is there. So this is where we can always be reliant on good science, and developing markers like CAS markers will help us to identify the differences between genotype and the phenotype and help us to clearly identify the even an unlabeled variety, what that gene might have been in that variety. So to get to the presence of RLM4 protecting RLM3, here's a classic example. These two varieties from location in Coronation, Alberta, if you know where that is. Again, work done during COVID. You can see that when RLM3 variety, this is in the same field, I should say, was alone, the pathogen was able to completely cause disease, whereas RLM4 was present with RLM3, the variety was excellent. So for some reason, RLM4 seems to be really a good gene that is protecting the RLM3 disease as well. So again, these are the reports that are developed by different seed testing laboratories for the farmer, and the farmer can then predict, the farmer can then make the right decision as to what variety they want to grow in the next season. So this is, again, I'm not going to spend time. If the slides are shared with you, you will be able to read these off. What I have mentioned is bringing all this information together is important as a scientist to give the best to the farmer. The best, at the end of the day, as I mentioned, the most important thing is to serve the farmer if you are an agriculture scientist. So the take-home messages is when high infection is present and our gene rotation with the right combination of genes is important than a crop rotation. So if farmers are going for a non-crop rotation, then our gene rotation has to be done in a high disease incident field. So is there good news? Of course, there's good news. There's new genes now coming along, and that means that there'll be new research coming along where you will have to now test how RLM2 is doing, RLM7 is doing, or other genes are doing. So it is always a very good opportunity for researchers to do research on this particular pathogen and learn more and more of the evolution of pathogens and how host pathogen interactions work. Now, to do that, the scientific community, global scientific community working on blackleg came together and published this article in 2022, mainly to give an opportunity for the breeders to quickly identify the R genes that are available in their genotypes. To do that, we sequenced almost 250 isolates, global isolates, And now these are available with all the researchers across the globe who can do this testing, and the breeders are at a hugely good advantage to use that. Now, what talk about fitness cost, there are certain pathogens that do not particularly like to stay in the virulent form. So, for example, AVRLM4 will rapidly change to the avirulent gene because there's a huge fitness cost, whereas AVRLM2 and 3 may not change that easily. So going back, this is work done in UK, and you can now see why that AVR-RLM4 protection might be really important in the array of genes that have been introduced to Canadian canola. So this is a very, again, very old information, but now with the new information that we have on our varieties, this might be very, very useful. So finally, you have to look at the pathogen diversity and work your methods. And when you look at the blackleg pathogen diversity, the gene flow is very high. So because of that, you need really R genes and quantitative resistance in multi-lines or regional deployment of these to make it a success story. Finally, I will like to end my presentation with a cartoon here showing that when there is a virulent allele, the RLM1 may not stand strong for a long time. It might break. But if the AVRL, the virulent allele, is trying to overcome many quantitative resistance genes, it may not be because they are standing together and being very strong. Now, if the best would be having the single gene resistance along with the quantitative resistance when the virulent allele will not be able to overcome either of those. And the virulent allele might fail because of that. So is it a perfect romance between canola and blackleg, or an evolutionary arms race? As plant pathologists, scientists, we really know that it is an evolutionary arms race. There's evolution of pathogen, and there is an important part for scientists to play to ensure sustainable canola production in Canadian canola and have the best market access, because we are a 90% export market driven industry, and strengthen the Canadian agriculture and our farmers. And be proud of the canola that was first developed in Canada at the University of Manitoba in the Department of Plant Science. And just a small plug here, as I mentioned, as editor-in-chief of Plant Pathology, I'll be very happy to entertain any articles that you might have in the future to be submitted to Plant Pathology or to the open access journal Plants, where also I am the founding editor-in-chief. And the work would never have been successful without the collaborations that I have developed over the years. And I've just only given the pictures of the Canadian researchers that were heavily involved with the research, and my longtime technician, Paula Parks, and to the many graduate students and postdocs that work in my lab and in other labs, as well as the collaborations with Australia, France, French researchers who freely shared their isolates and lines, and to the many funding organizations that believed in the research that we were doing to bring a difference to the Canadian agriculture with many new innovations and new methods being introduced. That's my lab that did most of that work at a particular time that's in 2022, and this is in 2023. A picture, the new crop of scientists in my lab who are now continuing to do the work that was started by others. And finally, my two sons seems to say there is a future in research innovation in evolution of fungal pathogens. Thank you to all of the evolution of fungal pathogen scientists who listen to this, especially to the graduate students. And I would welcome any questions either now or later through email if you have. Thank you.
Thank you so
Thank
much,
you.
Dr. Fernando. Are there any questions for Dr. Fernando?
Carol, I think there's one in the chat from Chris Blackman. If you want to read it, maybe it gets started the Q&A.
Okay, so Dr. Fernando, the question is, how long does a population of blackleg stick around in the field? Is 1 year R gene rotation generally enough to prevent blackleg from coming back before redeploying a cultivar?
Yeah, so it's an excellent question that is difficult to answer for the very reason that soils are different. And the stubble, I would say a fair bet would be 3-year rotation would be the best. The reason that I say that is because different soils contribute to different microbiomes, and these microbiomes will have an impact either in the survival of the pathogen or the detriment of the pathogen propagules, particularly the pseudothecia that will be destroyed by certain microbes. So we are also very interested in that angle, looking at the survival ability of the pathogen, particularly in combination with microbiomes that might be available in different soils, different conditions of the moisture, all that plays a role. But if I am to give an average number of years, I would say a 3-year rotation away from canola would be the ideal to reduce the inoculum to a level that is not going to be impactful to the farmer's bottom line.
Any other question?
I have a question. Can you hear me?
Good. Yeah.
Yes, yeah, go ahead.
Yeah, so very great talk. Nice to see put the research in the lab into perspective in terms of field work and as well as in reality what occurs in the field. So that's a really great talk. One question I have for you is regarding the mimicry that you observe, overcoming of the resistance. Do we know any thoughts on how the mechanism is? Is a common, for example, is overcoming RLM4, for example, escaping some kind of Gardaí, for example, for gene in the so-called R gene hypothesis? Do you know much about that in terms of mechanism?
The functional part of it is very little understood at this point, Gopal. So this is something that unfortunately not many of the people working on the blackleg pathosystem has taken on. It's an important area that is missing. Now that there are certain genes that have been cloned, I hope that there will be interest with scientists who are capable of doing it. I know that, Gopal, you are very good at doing that kind of functional analysis, but you are more focused on fusarium. So if there's people who are able to do this kind of research on the blackleg side, that will be really, really useful. This is a really good system to work with because of the gene-for-gene interaction being in play.
Any other question?
If there are questions that you feel like, you can always contact me also. That's my email address that I have there. Mari, how does it work now? We
Yeah,
go for
we can stop for about 5 minutes, and then the fellows that are interested can join you in a
Okay.
couple of minutes. So the fellows from EvoFunPath can have a little discussion with you.
Okay. Yeah. So thank you everyone for listening to me and participating. I really appreciate that.
Thanks, Dilantha.
Thank you, Gopal.
Thank you. See you in a couple of minutes.
Yeah.